Okay, thank you again, Mohsen. Yeah, so as you already mentioned that I'm a fourth year PhD student. So basically, I will be graduating soon. So my PhD major is environmental toxicology. So basically, my research is focusing on toxicogenomics of air toxic compound. So you also mentioned like aerosol. So lots of different air toxic compound and their effect into the human lung cell. And basically, I use different genomics to analyze their effect at the cellular level or genomics level. Uh, for my research, I use human epithelial lung cell and basically expose it to the different air toxic compound, for example, particulate matter, and it could be come from different traffic related particulate matter like gasoline exhaust, diesel exhaust. And eventually we generate some novel air toxic compound from our, chamber study, and we expose it and we see their effect. And yeah, so after basically finishing, as I mentioned, like I'm pretty much uh, done with my research, and basically in a stage like writing my dissertation. So yeah, so next stage will be maybe postdoc or industry. So yeah, I will be happy to answer you more details, like if you have any specific questions. Yeah,
Okay, thank you. So, are you like about to graduate this spring semester or in the next summer?
yeah,
What are you expecting?
by summer hopefully. Yeah.
Okay,
Mhm.
that's great.
Yeah.
So, I mean, like, little bit curious. Is this the same cell that COVID normally attach in like lung, like epithelial cells, or is little bit
Lung cell,
different?
yeah. So, I mean the lung epithelial cell. So it's basically BES2B, BES2B, the name of the cell. There are lots of different type of cells. Some cell is already carcinogenic, and eventually I am using normal cell. But my another, project is focusing on primary cell. So that means cells are collected from the human, directly from the human. So for that research, basically I'm focusing on healthy and COPD. COPD is basically chronic obstructive pulmonary diseases. So this healthy and disease two types of cell, and this is kind of primary and it's collected from the direct patients.
So, you do your normal exposure in the mice model, or
No. My study is
cell
solely
lines?
cell line. Yeah, no,
Okay.
not in any animal model. Yeah,
So like both cell line and human essence
human cell
models.
line. Exactly. Yeah.
That's that's cool. Yeah, I was reading your paper that you published. Uh, I forget the paper name in kind of environmental
Yes, and yeah,
that is,
environmental science and
yeah. But I just know that these are published by American Chemical Society, one of the
technology.
great
Yeah,
paper, and maybe people don't know about it. It's like a one of the like elite in chemistry
yeah.
field and toxicology field. So like I'm seeing like you have done lots of kind of DNA damage studies like P53 and how carcinogenic studies are going there and inflammations.
Mm-hmm.
So I'm actually also doing kind of some kind of stuff like though I do with chemotherapy,
Okay.
like how chemotherapy damage our normal cells and so I become interested on that particularly
Okay.
because I feel like this is kind of same work, but it's little bit environmental toxicology and it's like therapeutic
Exactly.
toxicity.
Yeah,
But
yeah.
that's really great because I see like the essays you do there, it's pretty much similar we do. So that's really nice.
Yeah. So the basic differences between the therapeutics and toxicology, basically, I'm exposing, toxic compound instead of drug. You know, so the idea is the same. So everything is the same. Whatever amount you're gonna expose, like the concentration, whatever study, dose dependent, time dependent, everything is the same. And then we expose it and see what is the effect of this specific toxic compound as like the drug. So if we wanna study any drug, we need to expose with the specific concentration and see what is their effect in terms of different, cellular and eventually their, effect on the genomics. And eventually, as you mentioned, like lots of them are going through the cellular pathways like p53 mediated stress response and this kind of stuff. And it can, if it cannot pass, it causes mutation, and ultimately the mutation leads to the cancer. So that's the phenomena, and eventually how they cause toxicity. So that's the important thing. And eventually, if you are kind of studying some drug that is working on the for example heart, so it can cause damage to the kidney, for example. So you need to know the toxicity. So toxicity is kind of interesting area to research. And so whatever the perspective, you can expose the toxic compound, you can study on the drug, or whatever your chemical of interest you can do. Yeah.
I think it's more people give more focus on in the field of drug development, because whatever we
Mhm.
are ingesting in our body, there should be any
Exactly.
kind of side effect and get
Exactly.
the end product going.
Yeah.
So yeah, I mean, you just tell a great thing about that, that it's depend on the time and also the dose. So I'm myself like struggling on that. So how actually you optimize those things? Like, is it like did you have any problem? Just you did one and it's worked, or you have to like little bit ups and down?
Yeah,
How is
yeah,
it
yeah. So
actually?
research cannot be just straightforward, and you cannot do just you cannot get done with a single try. You know that. Eventually,
Yeah.
everyone knows basically. You have some. You have to go through the trial and error. So basically, similar thing happened to me. Lots of things. So if you wanna culture the cells, and sometimes lots of factors, it can happen. Sometimes you cannot get your expected results, so you need to repeat. And eventually, the idea is basically my research is pretty straightforward. Like I need to select the dose. So for that study, I basically do dose dependent study. So different. So I basically select very high concentration, then mid-range, few mid-range, and then low, to see which dose is basically give me lower cytotoxicity, or at least below 30% of the cellular toxicity, cytotoxicity. So then after exposing that, basically I do basically RNA sequencing. I collect the RNA and this kind of stuff. So lots of steps that you need to repeat. And yeah,
So I seen you another work you did also on long non-coding RNAs. Uh, so
yeah.
how was that?
Okay, so this is basically a side project, during the COVID-19. So during the campus closure, so I basically collaborate with one of my uh, friend, my senior brother from Harvard University. So he's doing his postdoc there, and he's kind of expert in bioinformatics. So I have my RNA sequencing data. So what we did from the mRNA sequencing data, so we basically published based on the known gene, I mean the mRNA. So but we didn't look at the long non-coding RNA. This long non-coding RNA is important topic nowadays. So initially it was thought that the non-coding RNA is basically junk. So there is no any use. Only 2 to 3% of our whole genome. So only 2 to 3% is coding the
Coding genes.
co-coding gene and produce the protein. Other 98% or 97% DNA sequence or um, um, genomic part, they are basically non-coding RNA. So non-coding RNA, they have different types. So small, long non-coding RNA and long. Small means microRNA, and long means like at least it will be 200 nucleotide long. So recently, lots of study. So they
Yeah.
found that long non-coding RNA is regulating the gene expression. So from that perspective, because we found some interesting result from our research that after exposing our new lab generated aerosol, so basically we found lots of, as you mentioned, DNA damage, p53 mediated stress response, and inflammation. This kind of related pathway, they are basically perturbed. So we wanted to see that is there any long non-coding RNA they are contributing to this pathway, for example, DNA damage. And when we did the long non-coding RNA and mRNA co-expression, so we found lots of non-long non-coding RNA. They are related to DNA damage, p53 mediated pathway. And basically they are acting through cis and trans mechanism. Cis means like if they're you know closely if they act nearby genes, and trans means like if, for example, if you have a gene in the chromosome 1 and that long non-coding RNA works on this chromosome 1, I mean nearby genes, and for example if they work through the, for example, the long non-coding RNA is on chromosome 1 but they work on the chromosome 15 because they're distant in terms of location. So this is called trans, and we found some long non-coding RNA. They are basically regulating the gene expression,
That's
and
great.
sometimes and this is kind of interesting study. And that long non-coding RNA is known. So we basically did it through the bioinformatics tool, but this is kind of interesting. And that specific long non-coding RNA, it's been studied in the lab, that they are responsible for DNA damage repair. They're responsible for carcinogenesis. So that means the genes or mRNA we found, they already perturbed. They already they have a altered expression in the cellular system. But at the same time, there is another layer, that means long non-coding RNA. They are also regulating that specific genes or mRNA. So that's the interesting study. And maybe in future, we might change our statement that, okay, non-coding RNA is doing everything. This is my kind of thought that, they are basically regulating everything, every coding gene.
Yeah,
And
especially
yeah,
what
yeah,
I've learned, probably the mRNA stability is kind of regulated by that
exactly.
more.
MicroRNA. So they can degrade, and they degrade the mRNA, the microRNA, the small, around 20 to 25 nucleotide length. They're really, really small. And they can act as primer or something. They can easily bind with some RNA fragment and they can do whatever. So it's been well established in terms of microRNA, and long non-coding RNA kind of pretty new area. So we basically find out some long non-coding RNA, and we also found that this long non-coding RNA is being thought to be used as a potential biomarker, and early detection is necessary. For example, cancer. So it is a long process. So if you are in the late stage, then what will happen? So if you can detect some biomarker in terms of coding and non-coding um, genes or whatever, so you can at least see, okay, this kind of long non-coding RNA is being regulated or uh, altered, then maybe something is happening. So this is kind of future directional study that you mentioned, like long non-coding RNA and mRNA. But my current project, so pretty third project, I'm exposing diesel exhaust particle. So I'm gonna do the same. I will look at both mRNA level and long non-coding RNA, mRNA co-expression to see what kind of genes or non-coding RNA is basically responsible for diesel exhaust exposed, cells. Yeah.
So it's the benefit is that it give more detailed picture
Yeah.
of the of transcriptomics of the cells,
Transcriptomic. Yeah. Yeah. Both mRNA and
non-coding RNAs.
yeah, yeah.
That's
This is basically complete profiling.
a complete profile.
Yeah.
So I think your experience, your first project experience with RNA sequencing, give you that confidence to
Mm.
deal with
Yeah, yeah,
sequencing data, and that's why you
exactly.
The next step
Mhm.
with long non-coding RNA, you get the expertise from um, your colleague from the Harvard University, and that's make the third project more easy and
Yeah.
like
So we have a collaboration with other lab focusing on bioinformatics
yeah,
because
I really happy to see like I just before this used to heard that word that we need to collaborate. Why I didn't understand that much, but eventually like what I'm trying to do now in my PhD project. So my all first primary data come from my collaborative lab because they are doing something else in other
yeah.
tissues, and I actually work on the bone tissues. So I collected the
Mhm.
bone and almost got those all the samples free, and it like save many times of me. So it's like it's really good to do collaborate it.